Hello students, welcome back to my class. Dear students, in my last class I had discussed about the Mendel's law of inheritance, where I had taught you about Mendel's law, that is law of dominance and law of segregation. So in today's class I shall be teaching you new topic, that is incomplete dominance. So, dear students, if you remember that in my last class, I had discussed about dominant traits and recessive traits. Now, according to dominant traits, dominant alleles, they overpower the recessive trait, right? So, that is called your law of dominance. So where only dominant character used to express in the first generation, right? So what happened after the period of this Mendel's theory? Okay, so there are so many scientists, they came and they did experiment with different spaces, okay, different plant spaces, animal spaces. So one of them was Karl Correns, who was the first one who did an experiment with Mirabilis jalapas. So its common name is 4 o'clock plant. So here he had tried crossing a plant with the red flower and with white flower. Okay, and the result he found was different from what Mandel has said, or what Mandel experiment has said, okay, according to the Mandel's experiment. So what was that? Let us learn about this. So you will be understand better this with an example. So I will take an example from NCERT book, okay. So here in your book, a snapdragon, or you can say the dog plant has been taken, okay, dog flower, where this experiment has been carried out. So what happened was that two type of flower, or you can say a plant with two different flowers were taken, that is red flower and other is white. The red was the dominant, so it is expressed with allele capital R, and here on the other hand it is expresses with the small letter r. So the gamete came is capital R, and from here is your R small, that is lowercase r. Okay, now this is F1 generation. Now according to Mendel's law, what should be this? What should be the color of this? Red, right? But the actual result came was pink, not red. So then this phenomenon, incomplete dominance, was suggested, or it was given for this type of experiment, okay, for this type of character, where the dominant character, okay, they are not expressing their trait completely. The genes, they are not expressing the dominant trait completely, and that results in a blending of both the character of both the plants, okay, and this is called your incomplete dominance. So this was the result in F1 generation. Then this F1 generation, now they were self-crossed, okay, self-pollination takes place. So here the result came was both capital R. So here you can see, then this came your R. So the result according this is red. This should be red, right? But it is your pink. And this is your white. So according to this, phenotypic ratio is your 1:2:1, and the genotypic ratio as well, 1:2:1. Now in Mendel's experiment, we have seen that the phenotypic ratio and genotypic ratio wasn't same, okay, it was different. But here we can get to see the phenotypic ratio and genotypic ratio are similar, they are same. So this concept is said to be as your incomplete dominance, where uh, the dominant character, okay, they doesn't mask the recessive character completely, and as a result what happened, the dominant character and the recessive character they blend, okay, and the result come is different, okay, which is different from the Mandel's result. So I hope that incomplete dominance is clear to you. So if you have any doubts regarding this topic, then you can ask me, okay? Okay. So students, let us cover our next topic, okay? Now, I have been telling you about a dominant trait and a recessive trait, right? Now, the question is, how does these traits are expressed, okay? Now, this has been explained with an example. So, concept of dominance and recessive gene. Recessive traits. Now, first we should know that what is the role of gene? What does gene do? Gene is a unit of inheritance. I have told you, right? So it carries the information from one generation to the another generation. The traits, okay, the character which is expressed in a offspring is carried by the gene. So let us take an example which contain, okay, or which produce an enzyme, okay, and let us assume that this normal allele producing normal enzyme. So there is a substrate which required an enzyme, okay, and this normal allele will be producing the normal enzyme. So what happened? Two different alleles is produced, right? Modified alleles and unmodified alleles. Now, modified allele means when there are some changes, okay, when some changes takes place due to various reason, and one possible reason is mutation. So, what is mutation? We will learn this in details in our different chapter, okay? We will be learning this. Okay, so because of mutation, the alleles it get modified. The way you modified your bike, right? You bring some changes. You change the parts of bike, right? You modified it, isn't it? It become a little different from your original one, isn't it? Okay, I hope you could relate it. So modified allele and unmodified allele. So what happened? This two allele has some effect. They produce some hormone. Okay, so what happened? Modified allele are the normal alleles. Okay, they are the normal one. They are the original one, and they produce normal enzymes need for transformation of substrate S. Now I have said that the substrate, okay, S, it's required an enzyme for transformation, okay, it's required an enzyme for the transformation, and that is carried by normal allele. So unmodified allele means the normal allele, they are the normal alleles, okay, they doesn't get modified, okay, so they will produce the normal enzyme need for the transformation of the substrate. Say for an example, suppose this normal allele is carrying the information for the character tall, okay, tall trait. So this unmodified will remain the same. It means it will have the tall trait, okay, it will produce the normal allele which is required by the substrate, okay. But in modified allele, what happened? There are three cases. There is three possible cases that what they could produce. First one is either normal or less efficient. Less efficient means it will not overpower, or less effective. Okay, or normal you can say means normal or less efficient. It means that it is equal to the normal allele production. The same phenotype means normal means there is no changes, okay, they are producing the original one. The phenotype which carried by the normal allele will have this, okay. Second one is non-functional enzyme. So the enzyme they are producing is non-functional, means it is not functioning, okay, they are not showing any character, they are not expressing the character. Non-functional enzyme. See, they are carrying enzyme, right? Genes, they are carrying enzyme. So if it is non-functional, means they are not working, right? So there will be no change, there will be no changes and no enzyme is produced. Or there might be the case that where they are not producing any enzyme, correct? So this too becomes your recessive allele. See, where no character is expressed, where there is no function, no effective of any allele, okay, where any allele is not affecting the other character, that is called your recessive allele, okay. And the other one, the normal or the less efficient enzyme, it means it is equivalent to the normal allele, right? So if they are producing the normal allele, it means they are carrying the same phenotype, means the dominant character. If it is tall, if they are carrying tall character, then they will carry that only and there will be no change in them and they will express its character. So I hope that you are clear with the concept of dominance and recessive trait, that how does it is expressed in a spaces. So in my next class, I shall cover another topic. Okay, till then you just learn this topic and make a note on this. Learn the textbook. If you find any difficulty, just feel free to ask me. Okay, I'll try my best to help you out.